Mitosis is the division of one nucleus into two genetically identical nuclei, and the shortest stage in the cell cycle creates two diploid daughter cells, and is comprised of 5 stages: prophase, metaphase, metaphase, anaphase, and telophase. It is immediately followed by cytokinesis, the division of the cytoplasm. The mitotic phase alternates with a longer stage called interphase, which is made up of 3 subphases: the G1 phase, the S phase, and the G2 phase. During G2 of interphase, the stage that occurs prior to mitosis, a nuclear envelope encloses the nucleus, and the nucleus contains one or more nucleoli. A single centrosome, which are regions in animal cells that organize the microtubules of the spindle and contains 2 centrioles, duplicate into 2 centrosomes. Chromosomes duplicated during S phase cannot be seen individually because they haven't condensed. Chromatin, the sparse DNA strands, occupies the nucleus. During prophase, the chromatin fibers become more tightly coiled, condensing into discrete chromosomes. The nucleoli disappear. Each duplicated chromosome now appears as 2 identical sister chromatids joined at their centromeres. The mitotic spindle begins to form, which is composed of the centrosomes and the microtubules that extend from them. Radial arrays of shorter microtubules called asters extend from the centrosomes. The centrosomes begin to move away from each other. During prometaphase, the nuclear envelope fragments. The microtubules extending from each centrosome can now invade the nuclear area. The chromosomes have become even more condensed. Each of the 2 chromatids of each chromosome now has a kinetochore, a specialized protein structure at the centromere. Some of the microtubules attach to the kinetochores, becoming kinetochore microtubules, which jerk the chromosomes back and forth. Non-kinetochore microtubules interact with those from the opposite pole of the spindle. During metaphase, the longest stage of mitosis, the centrosomes are now at opposite poles of the cell. The chromosomes have all arrived at the metaphase plane, a plane that is equidistant between the spindle's 2 poles. The chromosome centromeres lie at the metaphase plane. For each chromosome, the kinetochores of the sister chromatids are attached to kinetochore microtubules coming from opposite poles. During anaphase, the shortest stage of mitosis begins when cohesin proteins are cleaved. This allows the 2 sister chromatids of each pair to part suddenly, each becoming a full-fledged chromosome. These 2 daughter chromosomes begin moving towards opposite ends of the cell as their kinetochore microtubules shorten. Because these microtubules are attached to the centromere region, the chromosomes move the centromere first. The cell elongates as the non-kinetochore microtubules lengthen. Now the 2 ends of the cell have equivalent and complete collections of chromosomes, or the diploid amount. During telophase, 2 daughter nuclei form in the cell. Nuclear envelope arise from the fragments of the parent cell nuclear envelope and other portions of the endomembrane system. The nucleoli reappear and the chromosomes have become less condensed. Mitosis, the division of one nucleus into 2 genetically identical nuclei, is now complete. During cytokinesis, the division of the cytoplasm is underway by late telophase, so 2 daughter cells appear shortly after the end of mitosis. In animal cells, cytokinesis involves the formation of a cleavage furrow, which pinches the cell in 2. In plant cells, during telophase, vesicles derived from the Golgi apparatus move along microtubules to the middle of the cell, where they coalesce, producing a cell plate. By the end of cytokinesis, the cell has produced 2 somatic diploid daughter cells.